Hey everybody, Ronnie here. So, I did a thing. I did this thing. I wrote a book. It's called How Evolution Works by Ronnie Joubert. This is a chunky boy. Okay, you can tell it's a big book. It's thick. It's about 500 and some odd pages. What we got? 525, 526. 526 pages. This is a labor of love. But what I did is I set out to write a book about how evolution works that was actually comprehensive, covered everything. I'm sure I missed something. It's still. But I'm putting this together here so that you can see it. This video is actually going to cover the first chapter, okay? Literally a read through of the first chapter, which is called The Origin of Life. If you like this video, give it a thumbs up, hit the subscribe button to see more content like this. And if you, and I'll put a link to the book down below that you can purchase on Amazon. It's available as an ebook for the Kindle or in this paperback form. And if you do click the link, I will get paid for your purchase. Thanks a lot. Enjoy the chapter 1, The Origin of Life. Part 1, Beginnings of Life. Chapter 1, The Origin of Life. Story, a primordial soup and the first spark of life. In the dim eons of Earth's infancy, the planet was a chaotic maelstrom of volcanic activity and tumultuous seeps. The surface was scarred by the relentless bombardment of meteorites, and the atmosphere was a noxious brew of gases that would be deadly to most life forms as we know them today. This was a world in its most primal state, a harsh and volatile environment that, paradoxically, set the stage for the emergence of life. Amidst the swirling chaos, the oceans were rich with a variety of simple chemicals, spewed from volcanic vents and carried by the rain into vast, warm seas. This primordial soup, as it would later be known, was a mixture of water, ammonia, methane, and hydrogen. The atmosphere above was dense with carbon dioxide, nitrogen, and water vapor, creating a thick haze that obscured the sun and trapped heat, maintaining the planet's surface temperature at a level conducive to chemical reactions. In this ancient world, lightning storms were frequent and intense, splitting the sky with jagged forks of electricity. These storms, combined with the intense ultraviolet radiation from a young sun, provided the energy necessary to drive chemical reactions. It was during one such storm, as lightning danced across the darkened sky and struck the surface of the ocean, that the first significant steps toward life began. Deep beneath the waves, in the mineral-rich waters near hydrothermal vents, a transformation was occurring. These vents, cracks in the ocean floor from which superheated water rich with minerals spewed forth, created pockets of extreme conditions. The intense heat and pressure, combined with the mineral-laden waters, provided a unique environment for chemical synthesis. In these dark and scalding depths, the primordial soup was being stirred by forces both violent and constant, creating the perfect conditions for the formation of complex, organic molecules. As the lightning struck the ocean's surface, it sent ripples of energy coursing through the water. Molecules collided and combined, breaking apart and reforming in myriad ways. Among these reactions, simple compounds began to coalesce into more complex structures. Amino acids, the building blocks of proteins, started to form. These molecules, though simple by today's biological standards, were a crucial step toward the complexity of life. In the shallow pools near the hydrothermal vents, another process was taking place. The repeated cycles of evaporation and condensation concentrated the organic molecules, promoting further reactions. As the waters evaporated, they left behind a concentrated solution of amino acids and other organic compounds. When the pools refilled with fresh mineral-laden water, the molecules had new opportunities to interact and form increasingly complex structures. One of the critical molecules that emerged from this primordial brew was RNA. Unlike DNA, which is the stable and reliable carrier of genetic information in most modern organisms, RNA is more versatile and can act as both the carrier of genetic information and a catalyst for chemical reactions. In the early Earth environment, RNA molecules began to form spontaneously, their sequences determined by the random assembly of nucleotides. These early RNA molecules were not yet capable of replication, but they laid the groundwork for the next critical step in the emergence of life. Through countless cycles of synthesis and breakdown, some RNA sequences began to fold into shapes that allowed them to catalyze their own replication. This self-replicating RNA was a significant milestone in the journey from chemistry to biology. In the warm, mineral-rich waters of the early Earth, the first protocells began to form. These were simple vesicles composed of lipid molecules, which naturally assembled into bilayers in water. These lipid bilayers enclosed small volumes of the primordial soup, creating microenvironments that concentrated the organic molecules and provided a rudimentary separation from the chaotic external environment. Inside these protocells, the self-replicating RNA molecules found a sanctuary where they could replicate more efficiently, protected from the harsh conditions outside. As the RNA molecules replicated within these protocells, they occasionally made errors, leading to variations in the RNA sequences. Some of these variations conferred advantages, allowing the protocells to grow and divide more effectively. Natural selection, a fundamental principle of evolution, began to take hold even at this molecular level. Protocells with more efficient RNA replicators outcompeted their less efficient counterparts, leading to the gradual increase in complexity and functionality. Over time, these early protocells became more sophisticated. The RNA molecules within them began to encode simple proteins, which further enhanced the protocell's functionality. These proteins acted as catalysts for various chemical reactions, increasing the efficiency of RNA replication and other metabolic processes. The protocells that could produce beneficial proteins had a significant advantage, leading to a feedback loop of increasing complexity. The transition from simple protocells to the first true cells was a gradual process, driven by the relentless forces of natural selection and chemical innovation. The lipid bilayers of the protocells became more stable and selective, allowing only certain molecules to pass through. This selective permeability was crucial for maintaining the internal environment necessary for life processes. As the first true cells emerged, they continued to evolve and diversify. Some cells developed the ability to harness the energy of sunlight through photosynthesis, using light-absorbing pigments to convert sunlight into chemical energy. This innovation had profound implications for life on Earth, as it allowed these early photosynthetic organisms to produce oxygen as a byproduct, gradually transforming the Earth's atmosphere and paving the way for the evolution of more complex life forms. In the oceans, these early cells formed vast colonies, mats, and stromatolites, creating complex ecosystems. These microbial communities interacted with their environment and with each other, forming intricate webs of ecological relationships. The exchange of genetic material through processes like horizontal gene transfer further accelerated the pace of evolution, allowing beneficial genes to spread rapidly through populations. As the atmosphere slowly accumulated oxygen from photosynthetic organisms, it reached a critical threshold that allowed for the evolution of aerobic respiration. This more efficient way of generating energy enabled the development of larger and more complex organisms. The stage was set for the evolution of multicellular life, leading to the rich tapestry of biodiversity we see on Earth today. The journey from a chaotic, lifeless planet to one teeming with life is a testament to the power of natural processes and the resilience of life's building blocks. The early Earth environment, with its volatile conditions and abundant chemical resources, provided the perfect crucible for the emergence of life. From the formation of simple organic molecules in the primordial soup to the evolution of complex cells capable of harnessing energy from their environment, the story of life's origins is one of remarkable ingenuity and adaptation. In the grand tapestry of evolutionary history, the origin of life is but the first chapter. It is a story that underscores the interconnectedness of all living things and the continuous thread of evolution that binds us to our ancient ancestors. As we look back on this primordial beginning, we gain a deeper appreciation for the complexity and beauty of life, and we are reminded of the incredible journey that has led to the diversity of life forms we see today. Concepts: abiogenesis, probiotic chemistry, abiogenesis. The origin of life, or abiogenesis, is the process by which living organisms arise naturally from non-living matter. This concept is foundational in understanding the emergence of life on Earth, setting the stage for the subsequent evolution of complex biological systems. Abiogenesis marks the transition from a purely chemical world to one dominated by biological processes, providing the first glimpse into the mechanisms that would eventually lead to the vast diversity of life we see today. Abiogenesis is fundamentally significant because it addresses the most profound question in biology: how did life begin? This inquiry delves into the conditions and processes that allowed for the assembly of organic molecules into structures capable of self-replication and metabolism. The study of abiogenesis seeks to uncover the initial steps that bridge the gap between non-living chemistry and living biology, offering insights into the nature of life itself and the potential for its existence elsewhere in the universe. Historically, the concept of abiogenesis has undergone significant evolution. In ancient times, spontaneous generation, the idea that life could arise from non-living matter spontaneously and continually, was a widely held belief. Aristotle, for instance, posited that simple organisms could arise from decaying matter, or mud. This notion persisted for centuries, with even eminent scientists like Isaac Newton and René Descartes subscribing to some form of spontaneous generation. However, the advent of the scientific method and the rise of experimental biology in the 17th and 18th centuries began to challenge these ideas. The work of Francesco Redi in the 1660s, who demonstrated that maggots on decaying meat came from eggs laid by flies and not from the meat itself, was an early blow to spontaneous generation. This was followed by Lazzaro Spallanzani's experiments in the 1760s, which showed that microorganisms did not appear in boiled broth unless exposed to air, further discrediting spontaneous generation. The definitive experiment that refuted spontaneous generation came in the mid-19th century with Louis Pasteur's work. Pasteur's meticulous experiments using swan neck flasks demonstrated that sterile nutrient broths remained free of microbial life unless exposed to microorganisms from the air. This conclusively disproved the idea of spontaneous generation for contemporary forms of life, but it left open the question of how life originally arose on the early Earth. The modern scientific approach to abiogenesis was significantly advanced by the groundbreaking work of Alexander Oparin and J.B.S. Haldane in the 1920s and 1930s. Independently, Oparin and Haldane proposed that early Earth had a reducing atmosphere, rich in molecules like methane, ammonia, hydrogen, and water vapor, but devoid of free oxygen. They hypothesized that energy from lightning, ultraviolet radiation, and volcanic activity could drive the formation of simple organic compounds from these gases. Over time, these compounds could accumulate in the oceans, forming a primordial soup where further chemical reactions could lead to the formation of more complex molecules and, eventually, the first living cells. This hypothesis was put to the test in the early 1950s by Stanley Miller and Harold Urey in a series of now famous experiments. Miller and Urey designed an apparatus that simulated the conditions of the early Earth as proposed by Oparin and Haldane. They filled a closed system with water, methane, ammonia, and hydrogen, and subjected the mixture to continuous electrical sparks to simulate lightning. After running the experiment for a week, they found that several amino acids, the building blocks of proteins, had formed spontaneously. The Miller-Urey experiment was a landmark achievement, providing the first experimental evidence that simple organic molecules necessary for life could form under prebiotic conditions. This work sparked a surge of interest in the field of prebiotic chemistry, leading to further experiments and theoretical developments aimed at understanding the chemical pathways that could lead to life. Subsequent research has expanded on the Miller-Urey experiment, exploring a variety of prebiotic conditions and energy sources. For instance, deep-sea hydrothermal vents have been proposed as potential sites for the origin of life. These vents provide a rich source of minerals and energy, creating microenvironments where complex organic molecules could form and accumulate. Laboratory simulations of vent conditions have shown that amino acids, nucleotides, and other organic molecules can indeed form under these conditions. Another line of investigation has focused on the role of minerals and surfaces in prebiotic chemistry. Certain minerals, such as clays and iron-sulfur compounds, can catalyze the formation of organic molecules and facilitate their assembly into larger structures. Experiments have demonstrated that these mineral surfaces can promote the formation of RNA and other biopolymers, suggesting a plausible pathway for the origin of life. The RNA world hypothesis is a central concept in modern theories of abiogenesis. This hypothesis proposes that RNA, rather than DNA or proteins, was the first molecule to carry genetic information and catalyze chemical reactions. RNA is unique in that it can store genetic information like DNA and act as a catalyst like proteins. The discovery of ribozymes, RNA molecules with catalytic activity, provided strong support for this hypothesis. Ribozymes could have facilitated the replication and diversification of early RNA molecules, leading to the evolution of increasingly complex molecular systems. One of the challenges in abiogenesis research is understanding how these early RNA molecules could have formed and replicated. While the spontaneous formation of RNA nucleotides under probiotic conditions has been demonstrated, the assembly of these nucleotides into long RNA chains remains a subject of intense study. Various mechanisms have been proposed, including the role of cyclic nucleotides, which are more easily polymerized, and the potential involvement of mineral surfaces in facilitating RNA assembly. Another key area of research is the transition from simple RNA-based systems to more complex cells. This involves understanding how protocells, simple vesicles with lipid bilayers, could have formed and incorporated RNA molecules. Protocells could have provided a protected environment for RNA replication and the evolution of metabolic pathways. Experiments have shown that fatty acids and other amphiphilic molecules can spontaneously form vesicles in water, supporting the plausibility of protocell formation under probiotic conditions. The study of abiogenesis also intersects with the search for life beyond Earth. Understanding the conditions and processes that led to the origin of life on Earth can inform the search for life on other planets and moons. For instance, the discovery of organic molecules and possible hydrothermal vent systems on moons like Europa and Enceladus raises the tantalizing possibility that similar processes could have led to the emergence of life elsewhere in the solar system. Modern theories of abiogenesis continue to evolve, incorporating new findings from chemistry, biology, and planetary science. The field is highly interdisciplinary, drawing on insights from a wide range of scientific disciplines. Researchers are continually refining their models of the early Earth environment, exploring new chemical pathways, and developing novel experimental techniques to test their hypotheses. One promising avenue of research involves the use of synthetic biology to recreate early life forms in the laboratory. By designing and constructing simple, self-replicating systems, scientists can gain insights into the minimal requirements for life and the pathways that could lead to increasing complexity. These synthetic systems can serve as models for understanding the origin of life and the principles that govern biological evolution. The study of abiogenesis is not just about understanding the past, it also has implications for the future. By uncovering the fundamental principles of life's origins, scientists can develop new technologies for biotechnology, medicine, and environmental science. For example, insights into the chemistry of early life could lead to the development of new catalysts and materials with applications in industry and healthcare. In conclusion, the study of abiogenesis is a profound and ongoing scientific endeavor that seeks to answer one of the most fundamental questions in biology: how did life begin? From the early hypotheses of spontaneous generation to the modern theories of probiotic chemistry and the RNA world, the field has made significant strides in understanding the processes that led to the emergence of life on Earth. The work of pioneers like Oparin, Haldane, Miller, and Urey has laid the foundation for a rich and dynamic field of research that continues to uncover the secrets of life's origins. Through a combination of experimental studies, theoretical modeling, and interdisciplinary collaboration, scientists are gradually piecing together the complex puzzle of abiogenesis, shedding light on the remarkable journey from chemistry to biology. As our knowledge of the early Earth environment and the chemical pathways that led to life grows, so too does our understanding of the potential for life elsewhere in the universe, offering exciting possibilities for future discoveries and technological advancements. Probiotic chemistry. Probiotic chemistry is the study of the chemical processes that occurred on the early Earth, leading to the formation of the first organic molecules and setting the stage for the emergence of life. Understanding these processes requires a multidisciplinary approach, drawing from chemistry, geology, atmospheric science, and biology to reconstruct the conditions and mechanisms that facilitated the transition from a lifeless planet to one teeming with life. This exploration begins with the primordial conditions of early Earth, which provided the necessary ingredients and environments for probiotic chemistry to unfold. The early Earth, approximately 4.5 billion years ago, was a vastly different place from the world we inhabit today. The planet's surface was molten, constantly reshaped by volcanic activity and frequent impacts from extraterrestrial objects. As the planet cooled, a solid crust began to form, and water vapor released from volcanic outgassing condensed to create the first oceans. These nascent bodies of water, interacting with the volcanic gases and minerals, formed a complex and dynamic system where probiotic chemistry could thrive. One of the key aspects of probiotic chemistry is the synthesis of simple organic molecules from inorganic precursors. The atmosphere of early Earth is thought to have been reducing, composed primarily of methane, ammonia, hydrogen, and water vapor, with little to no free oxygen. This composition was conducive to the formation of organic compounds, when energy was supplied in the form of ultraviolet radiation, lightning, or geothermal heat. In this environment, simple molecules such as hydrogen cyanide and formaldehyde could form through photochemical reactions and interactions between atmospheric gases. These molecules are highly reactive and serve as important building blocks for more complex organic compounds. For example, HCN can undergo polymerization to form adenine, one of the nucleobases essential for RNA and DNA. Similarly, formaldehyde can participate in reactions that lead to the formation of sugars, which are critical components of nucleic acids and metabolic pathways. The oceans played a crucial role in probiotic chemistry by providing a medium for these reactions to occur and for the accumulation of organic molecules. The interaction between atmospheric gases and the mineral-rich waters of the early oceans created a rich chemical environment. Rainwater, carrying dissolved gases and mineral salts, flowed into the oceans, concentrating these chemicals in various coastal and shallow marine environments. One of the most significant environments for prebiotic chemistry on early Earth was hydrothermal vents. These vents, located on the ocean floor, emit superheated water rich in dissolved minerals and gases. The discovery of modern hydrothermal vent ecosystems, teeming with life in the absence of sunlight, has provided crucial insights into the potential for these environments to support probiotic chemistry. The conditions at hydrothermal vents, including high temperatures, pressures, and the presence of catalytic minerals, create a unique setting for the synthesis and accumulation of organic molecules. At hydrothermal vents, the mixing of hot, mineral-laden water with cooler ocean water creates steep chemical gradients and provides a continuous source of energy. This energy drives a variety of chemical reactions, leading to the formation of molecules such as methane, ammonia, and hydrogen sulfide. These compounds can then participate in further reactions, leading to the synthesis of more complex organic molecules. One proposed mechanism for the formation of organic molecules at hydrothermal vents involves Fischer-Tropsch type synthesis. This process, which occurs on the surface of catalytic minerals such as iron and nickel, involves the hydrogenation of carbon monoxide and carbon dioxide to produce hydrocarbons and other organic compounds. Laboratory simulations of hydrothermal vent conditions have shown that FTT synthesis can indeed produce a variety of organic molecules, including hydrocarbons, alcohols, and carboxylic acids. Another important aspect of probiotic chemistry is the formation of amino acids, the building blocks of proteins. Amino acids can form through several pathways, including the Strecker synthesis, which involves the reaction of aldehydes or ketones with ammonia and hydrogen cyanide. Experiments simulating early Earth conditions have demonstrated that amino acids can form in significant quantities through these pathways. The presence of amino acids in meteorites, such as the Murchison meteorite, further supports the idea that these molecules could form under a variety of probiotic conditions. Nucleotides, the building blocks of nucleic acids, are another critical component of probiotic chemistry. Nucleotides are composed of a sugar, a phosphate group, and a nucleobase. The formation of nucleotides from simple organic molecules involves a series of complex reactions. One plausible pathway is the Formose reaction, which produces ribose and other sugars from formaldehyde. Ribose can then react with phosphate and nucleobases to form nucleotides. Experiments have shown that these reactions can occur under probiotic conditions, although the yields and efficiency of these processes remain areas of active research. The formation of membranes, which are essential for the compartmentalization of early life forms, is another crucial aspect of probiotic chemistry. Membranes are composed of lipid molecules that spontaneously form bilayers in water, creating enclosed vesicles. These vesicles, or protocells, provide a microenvironment where chemical reactions can occur in a controlled manner. The formation of lipid molecules from simpler precursors, such as fatty acids and glycerol, can occur through a variety of probiotic pathways. Laboratory experiments have demonstrated that fatty acids can form vesicles under a range of conditions, suggesting that protocells could have been an early step in the origin of life. In addition to hydrothermal vents, other environments on early Earth could have supported prebiotic chemistry. These include tidal pools, volcanic hot springs, and impact craters. Tidal pools, with their cycles of evaporation and refilling, could concentrate organic molecules and promote their polymerization. Volcanic hot springs, with their rich chemistry and thermal gradients, provide another potential setting for prebiotic reactions. Impact craters, formed by asteroid and comet collisions, would have generated intense heat and pressure, creating conditions for the synthesis of organic molecules. These craters could also serve as basins for the accumulation and concentration of prebiotic compounds. The role of mineral surfaces in prebiotic chemistry cannot be overstated. Minerals such as clays, iron-sulfur compounds, and phosphates can act as catalysts for the formation and polymerization of organic molecules. Clays, for example, have layered structures that can absorb and concentrate organic molecules, facilitating their interaction and polymerization. Experiments have shown that clays can catalyze the formation of RNA oligomers from activated nucleotides, providing a plausible pathway for the synthesis of nucleic acids. Iron-sulfur compounds, common in hydrothermal vent environments, can also catalyze a variety of prebiotic reactions. These minerals can facilitate the reduction of carbon dioxide to organic molecules, as well as the formation of peptides from amino acids. The catalytic properties of iron-sulfur minerals are thought to be similar to those of the active sites in many modern enzymes, suggesting a potential evolutionary link between prebiotic chemistry and biochemistry. Phosphates, which are essential for the formation of nucleotides and phospholipids, are another important component of prebiotic chemistry. The availability and concentration of phosphates on early Earth would have influenced the formation of these molecules. Studies have shown that certain minerals, such as schwabersite, a phosphate mineral found in meteorites, can release reactive phosphates under prebiotic conditions. These reactive phosphates can then participate in the formation of nucleotides and other phosphorylated compounds. The synthesis of complex organic molecules from simpler precursors is only part of the story of prebiotic chemistry. These molecules must also assemble into larger structures, such as polymers, that can carry out the functions necessary for life. Polymerization is the process by which monomers, or single units, are chemically bonded to form long chains or networks. In the context of prebiotic chemistry, polymerization is crucial for the formation of proteins, nucleic acids, and polysaccharides. One of the challenges in prebiotic chemistry is understanding how polymerization could occur under early Earth conditions. The formation of long, stable polymers typically requires energy input and specific conditions. However, prebiotic environments could provide the necessary energy and conditions for these reactions. For example, the drying and rehydration cycles in tidal pools can promote the condensation reactions needed to form peptides from amino acids. Similarly, the thermal gradients and catalytic surfaces at hydrothermal vents can facilitate the polymerization of nucleotides into RNA. The self-assembly of lipid molecules into vesicles, or protocells, is another key aspect of prebiotic chemistry. Protocells provide a compartmentalized environment where chemical reactions can occur more efficiently and where molecules can be protected from the external environment. The formation of protocells from simple lipid molecules is a spontaneous process driven by hydrophobic interactions. When lipids are dispersed in water, they spontaneously form bilayers that enclose small volumes of water, creating vesicles. These vesicles can encapsulate organic molecules and provide a microenvironment for further chemical reactions. One of the critical steps in the origin of life is the emergence of self-replicating systems. Self-replication is the process by which molecules can produce copies of themselves, a fundamental property of life. In prebiotic chemistry, self-replication could have been driven by the autocatalytic properties of certain molecules. For example, RNA molecules can catalyze their own replication through base pairing interactions. Ribozymes, or catalytic RNA molecules, can facilitate the formation of complementary RNA strands, leading to the replication of the original RNA molecule. The evolution of self-replicating systems from simple organic molecules is a complex and poorly understood process. However, studies of autocatalytic networks and chemical systems have provided insights into how self-replication could have emerged. These systems involve sets of molecules that catalyze each other's formation, creating a network of reactions that can sustain and propagate itself. Such autocatalytic networks could have been a precursor to the more complex self-replicating systems seen in living organisms. The transition from chemical systems to biological systems involves not only the formation of self-replicating molecules, but also the development of metabolic pathways. Metabolism is the set of chemical reactions that occur within a cell to maintain life, including the conversion of energy, the synthesis of biomolecules, and the regulation of cellular processes. The emergence of metabolic pathways in prebiotic chemistry would have been driven by the availability of chemical energy and the catalytic properties of minerals and organic molecules. One proposed model for the origin of metabolism is the metabolism first hypothesis, which suggests that simple metabolic pathways could have evolved before the emergence of genetic information. This model posits that networks of chemical reactions could have formed in prebiotic environments, driven by the availability of energy and catalytic surfaces. These reaction networks could generate complex molecules and provide the energy necessary for the emergence of self-replicating systems. In contrast, the genetics first hypothesis suggests that self-replicating genetic molecules, such as RNA, emerged first and then directed the formation of metabolic pathways. This model is supported by the RNA world hypothesis, which proposes that RNA molecules were the first to carry genetic information and catalyze chemical reactions. In this scenario, the evolution of RNA-based life forms would have driven the development of metabolic pathways to support replication and growth. Both models have their strengths and weaknesses, and it is likely that the origin of life involved a combination of both metabolic and genetic processes. The interplay between self-replicating molecules and metabolic networks could have created a feedback loop, driving the evolution of increasingly complex systems. For example, the replication of RNA molecules could generate mutations and variations, leading to the evolution of ribozymes with new catalytic activities. These ribozymes could then catalyze new metabolic reactions, creating a dynamic and evolving system. The study of prebiotic chemistry is an ongoing and dynamic field, with new discoveries and insights continually reshaping our understanding of the origin of life. Advances in experimental techniques, computational modeling, and interdisciplinary collaboration are driving progress in this area. For example, modern analytical tools such as mass spectrometry and nuclear magnetic resonance spectroscopy allow scientists to characterize complex mixtures of organic molecules and trace the pathways of chemical reactions in prebiotic systems. Computational modeling and simulations are also playing a crucial role in prebiotic chemistry research. These tools allow scientists to explore the vast parameter space of prebiotic conditions, testing different scenarios and reaction pathways. By modeling the interactions between molecules, minerals, and environmental conditions, researchers can generate hypotheses and guide experimental studies. Interdisciplinary collaboration is essential for advancing our understanding of prebiotic chemistry. Chemists, geologists, biologists, and planetary scientists work together to reconstruct the conditions of early Earth, identify potential prebiotic environments, and design experiments to test hypotheses. This collaborative approach is helping to bridge the gap between chemistry and biology, shedding light on the processes that led to the emergence of life. In conclusion, prebiotic chemistry is a foundational aspect of understanding the origin of life. The chemical processes that occurred on the early Earth, including the formation of simple organic molecules, the role of hydrothermal vents, and other environments, set the stage for the emergence of self-replicating systems and metabolic networks. These processes were driven by the unique conditions of the early Earth, including the reducing atmosphere, the presence of water and minerals, and the availability of energy sources such as lightning and geothermal heat. Through a combination of experimental studies, computational modeling, and interdisciplinary collaboration, scientists are gradually unraveling the complex pathways that led to the transition from a lifeless planet to one teeming with life. This research not only enhances our understanding of our origins, but also informs the search for life beyond Earth and the potential for life in diverse environments across the universe. Conclusion: the study of abiogenesis and prebiotic chemistry is an exploration into the profound mystery of how life originated on Earth. The journey begins with understanding the conditions on the primordial Earth, a planet vastly different from the one we know today. This ancient world, characterized by a reducing atmosphere and a volatile surface, provided a fertile ground for the birth of organic molecules, the fundamental building blocks of life. Abiogenesis, the process by which life arises naturally from non-living matter, is a cornerstone of biological sciences. Its significance lies not just in explaining the origin of life on Earth, but also in offering insights into the potential for life elsewhere in the universe. The early Earth's atmosphere, dominated by methane, ammonia, hydrogen, and water vapor, set the stage for complex chemical reactions driven by energy sources such as ultraviolet radiation, lightning, and geothermal heat. These reactions, occurring in the oceans and other water bodies, led to the formation of simple organic molecules. Historically, the concept of abiogenesis has evolved significantly. Ancient beliefs in spontaneous generation gave way to scientific scrutiny in the 17th and 18th centuries, culminating in Louis Pasteur's definitive experiments that refuted spontaneous generation for existing life forms. The 20th century saw the development of modern abiogenesis theories, particularly the hypothesis proposed by Alexander Oparin and J.B.S. Haldane. They envisioned early Earth as a chemically rich environment where simple molecules could combine to form complex organic compounds, creating a primordial soup from which life could emerge. The groundbreaking Miller-Urey experiment in the 1950s provided empirical support for this hypothesis. By simulating early Earth conditions, Stanley Miller and Harold Urey demonstrated that amino acids, the building blocks of proteins, could form spontaneously. This experiment marked a pivotal moment in the study of the origin of life, showing that the precursors of life could indeed arise from non-living chemical processes. Prebiotic chemistry delves deeper into the specific chemical processes that occurred on early Earth. The reducing atmosphere played a crucial role in these processes, facilitating the formation of molecules like hydrogen cyanide and formaldehyde. These molecules are highly reactive and serve as essential precursors for more complex organic compounds. For example, hydrogen cyanide can polymerize to form adenine, a fundamental component of nucleic acids, while formaldehyde can participate in reactions that produce sugars, critical for RNA and DNA formation. The role of water bodies, particularly the oceans, in prebiotic chemistry is paramount. The interaction between atmospheric gases and mineral-rich waters created a dynamic chemical environment conducive to the synthesis of organic molecules. Hydrothermal vents, with their unique combination of heat, pressure, and mineral content, provided ideal conditions for these reactions. These vents emit superheated, mineral-laden water, creating steep chemical gradients and a continuous source of energy, driving the synthesis of organic molecules. Hydrothermal vent environments likely facilitated Fischer-Tropsch-type synthesis, a process that produces hydrocarbons and other organic compounds from carbon monoxide and hydrogen. This mechanism, occurring on the surfaces of catalytic minerals like iron and nickel, illustrates how the unique conditions at hydrothermal vents could have supported the formation of life's building blocks. Additionally, these environments could have supported the synthesis of amino acids and nucleotides, crucial components of proteins and nucleic acids. Minerals and surfaces played a critical role in prebiotic chemistry. Clays and iron-sulfur compounds, abundant in early Earth environments, acted as catalysts for the formation and polymerization of organic molecules. Clays, with their layered structures, could absorb and concentrate organic molecules, facilitating their interaction and polymerization. Iron-sulfur minerals, common in hydrothermal vent environments, could catalyze the reduction of carbon dioxide to organic molecules and the formation of peptides from amino acids. These catalytic properties of minerals suggest an evolutionary link between prebiotic chemistry and modern biochemistry. Phosphates are another essential component of prebiotic chemistry, crucial for the formation of nucleotides and phospholipids. The availability of reactive phosphates, possibly from minerals like schreibersite found in meteorites, would have influenced the formation of these molecules. Reactive phosphates can participate in the formation of nucleotides, which are the building blocks of nucleic acids, and in the synthesis of phospholipids, which form cell membranes. The polymerization of simple organic molecules into larger structures is a critical step in the origin of life. This process, essential for the formation of proteins, nucleic acids, and polysaccharides, requires specific conditions and energy inputs. Prebiotic environments, such as tidal pools and hydrothermal vents, provided these conditions. For example, the drying and rehydration cycles in tidal pools could promote the condensation reactions needed to form peptides from amino acids. Similarly, the thermal gradients and catalytic surfaces at hydrothermal vents could facilitate the polymerization of nucleotides into RNA. The self-assembly of lipid molecules into vesicles, or protocells, is another crucial aspect of prebiotic chemistry. These protocells provide a microenvironment where chemical reactions can occur more efficiently and where molecules can be protected from the external environment. The formation of lipid bilayers from simple lipid molecules, driven by hydrophobic interactions, is a spontaneous process. These bilayers enclose small volumes of water, creating vesicles that can encapsulate organic molecules and provide a microenvironment for further chemical reactions. The emergence of self-replicating systems marks a pivotal point in the transition from chemistry to biology. Self-replication, the ability of molecules to produce copies of themselves, is a fundamental property of life. In prebiotic chemistry, self-replication could have been driven by the autocatalytic properties of certain molecules. For example, RNA molecules can catalyze their own replication through base pairing interactions. Ribozymes, or catalytic RNA molecules, can facilitate the formation of complementary RNA strands, leading to the replication of the original RNA molecule. The evolution of self-replicating systems from simple organic molecules is a complex and poorly understood process. However, studies of autocatalytic networks and chemical systems provide insights into how self-replication could have emerged. These systems involve sets of molecules that catalyze each other's formation, creating a network of reactions that can sustain and propagate itself. Such autocatalytic networks could have been precursors to the more complex self-replicating systems seen in living organisms. The transition from chemical systems to biological systems involves not only the formation of self-replicating molecules, but also the development of metabolic pathways. Metabolism is the set of chemical reactions that occur within a cell to maintain life, including the conversion of energy, the synthesis of biomolecules, and the regulation of cellular processes. The emergence of metabolic pathways in prebiotic chemistry would have been driven by the availability of chemical energy and the catalytic properties of minerals and organic molecules. One proposed model for the origin of metabolism is the metabolism first hypothesis, which suggests that simple metabolic pathways could have evolved before the emergence of genetic information. This model posits that networks of chemical reactions could have formed in prebiotic environments, driven by the availability of energy in catalytic surfaces. These reaction networks could generate complex molecules and provide the energy necessary for the emergence of self-replicating systems. In contrast, the genetics first hypothesis suggests that self-replicating genetic molecules, such as RNA, emerged first and then directed the formation of metabolic pathways. This model is supported by the RNA world hypothesis, which proposes that RNA molecules were the first to carry genetic information and catalyze chemical reactions. In this scenario, the evolution of RNA-based life forms would have driven the development of metabolic pathways to support replication and growth. Both models have their strengths and weaknesses, and it is likely that the origin of life involved a combination of both metabolic and genetic processes. The interplay between self-replicating molecules and metabolic networks could have created a feedback loop, driving the evolution of increasingly complex systems. For example, the replication of RNA molecules could generate mutations and variations, leading to the evolution of ribozymes with new catalytic activities. These ribozymes could then catalyze new metabolic reactions, creating a dynamic and evolving system. The study of prebiotic chemistry is an ongoing and dynamic field, with new discoveries and insights continually reshaping our understanding of the origin of life. Advances in experimental techniques, computational modeling, and interdisciplinary collaboration are driving progress in this area. For example, modern analytical tools such as mass spectrometry and nuclear magnetic resonance spectroscopy allow scientists to characterize complex mixtures of organic molecules and trace the pathways of chemical reactions in prebiotic systems. Computational modeling and simulations are also playing a crucial role in prebiotic chemistry research. These tools allow scientists to explore the vast parameter space of prebiotic conditions, testing different scenarios and reaction pathways. By modeling the interactions between molecules, minerals, and environmental conditions, researchers can generate hypotheses and guide experimental studies. Interdisciplinary collaboration is essential for advancing our understanding of prebiotic chemistry. Chemists, geologists, biologists, and planetary scientists work together to reconstruct the conditions of early Earth, identify potential prebiotic environments, and design experiments to test hypotheses. This collaborative approach is helping to bridge the gap between chemistry and biology, shedding light on the processes that led to the emergence of life. In conclusion, the study of abiogenesis and prebiotic chemistry is a profound and ongoing scientific endeavor that seeks to answer one of the most fundamental questions in biology: how did life begin? From the early hypotheses of spontaneous generation to the modern theories of prebiotic chemistry and the RNA world, the field has made significant strides in understanding the processes that led to the emergence of life on Earth. The work of pioneers like Oparin, Haldane, Miller, and Urey has laid the foundation for a rich and dynamic field of research that continues to uncover the secrets of life's origins. Through a combination of experimental studies, computational modeling, and interdisciplinary collaboration. Scientists are gradually piecing together the complex puzzle of abiogenesis, shedding light on the remarkable journey from chemistry to biology. As our knowledge of the early Earth environment and the chemical pathways that led to life grows, so too does our understanding of the potential for life elsewhere in the universe, offering exciting possibilities for future discoveries and technological advancements. As we transition to the next chapter, we will delve deeper into the formation of the first molecules capable of self-replication, setting the stage for the emergence of life. This exploration will take us from the primordial soup of simple organic molecules to the complex networks of interactions that characterize living systems. We will examine the mechanisms by which these molecules could have formed, replicated, and evolved, paving the way for the development of cellular life. Through this journey, we will gain a deeper appreciation for the intricate and dynamic processes that underpin the origin and evolution of life on Earth.